Welcome to the deep dive. Today, we're going to be looking at a research article from 2001.
Okay.
It's from Chromatographia
Yeah.
by Sweeney, Wong, and Shalliker.
Interesting.
And we're going to be looking at this idea of how you separate different forms of the same polymer
That's fun.
and why that even matters.
Yeah.
So get ready,
Okay.
because we're going to uncover how this seemingly obscure area of chemistry has huge implications for things like medicine, the environment, and maybe even just the everyday stuff you use and don't even realize.
That's true.
Yeah.
You know, we're so used to polymers being everywhere.
Right.
We rarely think about the fact that even tiny changes to how those molecules are arranged can just completely change a material's properties.
Yeah.
So the research we're looking at
Yeah.
focuses on polystyrene.
Okay.
Think like disposable cups, packaging peanuts, that kind of stuff.
Packing peanuts.
And it really digs into the challenge of separating its different isomers.
Okay.
Yeah.
I am intrigued. But before we get into the specifics of the research,
Okay.
can we back up a bit?
Sure.
I remember enough from high school chemistry to know that isomers have the same chemical formula,
Right.
but their atoms are arranged differently.
Exactly.
But I definitely need a refresher on how that actually plays out with polymers,
Okay.
especially when it comes to something like polystyrene.
Absolutely.
Yeah.
So let's start with chirality.
Okay.
Think of it like your hands.
Oh.
They're mirror images,
Mm-hmm.
but you can't perfectly stack them on top of each other.
Okay.
That's because they're chiral.
Okay.
Now, molecules can be chiral too,
Uh-huh.
and those mirror images are called enantiomers.
Okay.
And even though they have the same atoms,
Okay.
enantiomers can behave very differently,
Uh-huh.
especially when they interact with biological systems.
Right.
Remember the thalidomide tragedy?
Oh, yeah.
Yeah?
It's a chilling example.
Yeah.
One enantiomer of thalidomide was effective against morning sickness,
Yeah.
but the other one caused devastating birth defects.
Precisely.
Yeah.
And that's a stark reminder of why understanding and separating different isomers is so crucial.
Right.
Yeah. But with polymers, things get even more complex.
Okay.
Because you have this long chain of repeating units.
Uh-huh.
So on top of chirality,
Okay.
you have to consider how those units are arranged along the chain.
Okay.
And that's what we call tacticity.
So you're saying that even if we ignore chirality for a second,
Uh-huh.
just the way that the monomers are linked together
Yeah,
can create different forms of polystyrene.
exactly.
Wow.
Think of building a tower with Legos.
Okay.
If all the bricks are lined up perfectly on the same side,
Uh-huh.
you get a strong, rigid structure
Okay.
that's like an isotactic polymer,
Okay.
very ordered.
Uh-huh.
But if you alternate the bricks on each side,
Okay.
it's a bit more flexible,
All right.
but still pretty organized.
Oh.
That's syndiotactic.
Okay.
Then imagine just throwing the bricks together randomly. That's a tactic.
Okay.
And as you can imagine, that's going to be the weakest and the least structured of the three.
Okay, that analogy really helps.
Sure.
So we've got different isomers due to chirality.
Yeah.
And then on top of that, we have the different tacticities.
Mm-hmm.
No wonder separating these various forms of polystyrene is such a challenge.
Right.
Wow.
It's like trying to sort a mountain of Legos, not just by color, but also by how they're connected.
Yeah.
And that's where Sweeney, Wong, and Shalliker come in.
Okay.
Their research in chromatography focused on using a technique
Okay.
called reversed phase high performance liquid chromatography.
Okay. Or PHPLC.
All right. RPHPLC.
Yeah.
That sounds pretty technical.
It is a little bit.
Can we break it down a bit?
Sure.
I know chromatography is used to separate mixtures,
Yeah.
but how does it actually work?
Okay.
Especially when you're dealing with something as complex as polymer isomers.
So imagine you have a long, thin tube
Okay.
packed with a material that has specific chemical properties.
And that's your stationary phase.
Right.
Then you pump your mixture of polystyrene molecules dissolved in a liquid through this tube.
Okay.
And that liquid is the mobile phase.
So the polystyrene molecules are basically racing through this tube.
In a way, yes.
Okay.
But as they travel through that stationary phase,
Uh-huh.
some molecules will interact with it more strongly than others.
Okay.
And those interactions are based on things like the size of the molecule,
Okay.
its shape,
Uh-huh.
and its chemical properties,
Right.
which, as we've been discussing, are all affected by chirality and tacticity.
So those interactions are what cause the different isomers to separate.
Exactly.
Then they'll get slowed down to different degrees
Yeah.
depending on how much they're attracted to that stationary phase.
You got it.
Okay.
The key is that you need a stationary phase that can recognize even the subtle differences between the isomers.
Right.
So Sweeney, Wong, and Shalliker compared two different stationary phases.
Okay.
A traditional C18 column and something called a carbon-clad zirconia column.
Okay.
And this is where the results get really interesting.
Okay, I'm on the edge of my seat.
All right.
Tell me more about these columns and what they found. What's so special about this carbon-clad zirconia?
Well, that's exactly what we'll delve into next.
Sounds like it's time for part 2 of this deep dive.
I think so. Welcome back to the deep dive. Let's continue exploring how Sweeney, Wong, and Shalliker separated polystyrene isomers using different types of chromatography.
All right, you left us hanging with the C18 and carbon-clad zirconia columns.
Oh, right.
What happened when they actually ran the polystyrene through these columns?
Well, they started with the C18 column,
Okay.
which is kind of the gold standard in this type of chromatography.
Yeah.
And it did a decent job of separating the polystyrene molecules based on their size or molecular weight.
Okay.
So smaller chains zip through faster,
Okay.
while the bigger ones took their time.
Makes sense. But what about separating the isomers? Did the C18 column differentiate between the
Yeah.
isotactic, syndiotactic, and atactic forms?
That's where the C18 fell a little bit short.
Okay.
While it could sort by size,
Mhm.
it couldn't really distinguish those subtle structural differences between the isomers. You know, it's
Okay.
like trying to separate grains of sand based on tiny variations in their shape.
Right.
Almost impossible.
So the C18 was good for a rough separation, but not for that fine-grained analysis they needed to isolate specific isomers.
Exactly. And that's where the carbon-clad zirconia column really shines.
Okay.
When they ran the polystyrene through this column,
Mhm.
they saw a much more detailed separation.
Okay.
It's like suddenly having a high-definition picture instead of a blurry one.
That's fascinating.
Yeah.
What is it about the carbon-clad zirconia that makes it so good at separating isomers?
It comes down to the surface chemistry.
Okay.
The carbon-clad zirconia has this unique surface that allows it to interact with the polystyrene molecules in a much more nuanced way. It's not just about size anymore,
Right.
but also about the specific arrangement of those styrene units.
Okay.
The tacticity and chirality we talked about earlier.
So the carbon-clad zirconia is like a molecular detective,
Exactly.
able to pick up on those tiny clues that the C18 misses.
Yeah.
Okay.
Sweeney, Wong, and Shalliker found that this column could even separate isomers that have the same number of styrene units, but with those units arranged differently.
Wow, that's some serious separation power.
Yeah.
But why should we care about this?
Right.
You mentioned this research having real-world applications.
Yeah.
Can you give me some examples?
Let's go back to the thalidomide tragedy.
Okay.
Imagine if back then they had the ability to easily separate those two enantiomers.
Right.
We might have avoided that disaster altogether.
That's a powerful point.
Yeah,
So being able to separate isomers has huge implications for drug development and safety.
absolutely. And it goes beyond just pharmaceuticals.
Okay.
Think about microplastics.
Okay.
We know they're a huge environmental problem, but different types of plastics break down differently. Being able to analyze the composition of microplastics, maybe even at the isomer level,
Right.
could help us understand how they're impacting the environment and
Right.
develop better ways to deal with them.
Okay, that's blowing my mind a little.
Yeah.
So we've gone from separating polystyrene isomers in a lab
Yeah.
to potentially solving major problems in medicine and the environment.
Uh-huh.
That's quite a leap.
And there's more.
Okay.
This research also has implications for material science.
Okay, how so?
Imagine being able to fine-tune the properties of plastics by controlling the arrangement of their molecules.
Wow.
We could design materials with exactly the strength, flexibility, and even biodegradability we need for specific applications.
This is truly a deep dive.
Yeah,
And it seems like we've only scratched the surface of the potential applications of this research.
I think so.
Is there anything else you want to share about the findings of Sweeney, Wong, and Shalliker, or about other applications?
There are a few additional points we should cover to bring it all together.
Welcome back to the deep dive. So far, we've looked at how these tiny little differences in polymer structure can lead to huge changes in material properties.
Yeah.
And we've also seen how Sweeney, Wong, and Shalliker use chromatography to separate polystyrene isomers with the carbon-clad zirconia column, proving to be particularly effective.
Yeah, it was great.
Now let's dive deeper into the real-world applications of this research.
So we've touched upon the implications for drug development, microplastics analysis, and materials science. But let's unpack those a bit further.
Okay, starting with drug development, the thalidomide example really hit home.
Yeah.
Being able to isolate specific enantiomers could prevent similar tragedies from happening in the future.
That's right. And it's not just about avoiding harmful side effects.
Right.
By separating enantiomers, we can also potentially enhance drug efficacy. Sometimes only one enantiomer is responsible for the desired therapeutic effect. So by isolating it, we could create more targeted and potent medications.
That makes sense. So this research isn't just about making drugs safer,
Right.
but potentially making them more effective as well.
Exactly. Now let's shift gears to microplastics.
Okay.
As we've discussed, these tiny plastic fragments are a growing environmental concern.
Right.
But figuring out their impact is a challenge. Different types of plastic, even different isomers of the same plastic might break down in different ways.
So being able to analyze the composition of microplastics at a very detailed level, even down to the isomer level, could be crucial.
Absolutely. Imagine being able to track not just how much microplastic is present, but what specific types are accumulating in different environments or even in organisms.
Right.
This could help us pinpoint the most problematic plastics and develop more effective solutions. And Sweeney, Wong, and Shalliker's work with chromatography gives us a tool to do just that.
It's amazing to think that a technique designed to separate polystyrene isomers in a lab could have such a big impact on how we tackle global pollution.
And then there's material science.
Okay.
The ability to control the arrangement of molecules within a polymer opens up a world of possibilities for creating new materials. We could tailor plastics to have specific strengths, flexibilities, and melting points.
So we're talking about designing plastics for very specific purposes.
Right.
Rather than just relying on the general properties of a particular polymer.
Precisely. Imagine biodegradable plastics that break down quickly in the environment.
Okay.
Or super strong plastics that can replace metal in certain applications. By controlling tacticity, we can even influence how a plastic interacts with light, heat, or electricity.
It seems like this research has the potential to revolutionize the way we think about and use plastics.
Yeah.
It's not just about the polymer itself.
Right.
But about how we control its structure at a molecular level to create materials with entirely new properties.
That's the key takeaway here.
Okay.
Sweeney, Wong, and Shalliker's work on separating polystyrene isomers wasn't just an academic exercise. It provided a tool, a new way of looking at and manipulating polymers with applications that go far beyond the initial research.
It's a beautiful example of how fundamental science can lead to unexpected and impactful innovations.
Definitely.
It really makes you appreciate the power of curiosity and the importance of supporting research that pushes the boundaries of our understanding.
Indeed. And who knows what other discoveries await as we continue to delve deeper into the intricate world of polymers.
Thanks for joining us on this deep dive into the fascinating world of polymer separation.
Of course.
We hope you've gained a new appreciation for the complexity of these molecules and the incredible potential they hold for shaping our future.